Hi, folks. Thank you for joining me for another lecture. This time we're going to be diving into some key points about genetics and inheritance or heritability of traits. How do traits get passed on from parents to offspring? What determines the traits that are going to be expressed in those offspring? Those types of questions. Now, I want to make sure everyone is aware that in the module for this week, there's a handout posted. It's called Human Reproduction and Heredity Handout. So look at that handout. You can either look at that now before getting started with this lecture, or make sure you go back to that after this lecture and read some of the details about some explanations of what we're doing and going over in this lecture video. So that handout is just meant to be a supplement to put some of the ideas together on one page. It may not be necessary for you if you can slow this lecture down and get each of the points as we go through them here. I put a picture of a pea plant here because pea plants are going to feature heavily in this lecture. There is the father of genetics and heredity, the person who first kind of elucidated these relationships among traits in parents and traits in their offspring and how this information gets passed down. His name was Gregor Mendel, and he worked with pea plants a lot. So you'll see some references to various traits of pea plants. This is the flower of a pea plant. Here's a drawing of Mendel working in his garden. Gregor Mendel was an Augustinian monk living in Austria, and he did most of this experimentation and groundbreaking work in the mid-1800s. This was actually just after Darwin put out his book on the origin of species by means of natural selection. When Darwin first proposed evolution of species and a mechanism for that evolution being natural selection, Darwin knew that individuals inherited traits from their ancestors, from their parents, but Darwin did not have an idea of how those traits were inherited. Certainly, he didn't know anything about a gene or different versions of genes and what's going on at a molecular level for these chromosomes and genes to be passed down to the next generation. There was very little known about genetics at that time. So people knew that it happened, that offspring inherited traits from their parents, but they didn't know how it happened. And Gregor Mendel was an absolutely fastidious scholar. He toiled away in his garden and he didn't seek fame. He was a pretty low-key guy, in fact, but a very determined guy who just did these experiments and kept doing more and more experiments with pea plants and had such a large data set that he was able to look at all of his data, and by viewing these outcomes at a very macroscopic level, something he could easily observe, he was able to deduce what was going on with respect to the genes of these plants. So let's have a look at what Mendel did. This work wasn't really acknowledged until much later because it was hidden away in his notebooks and obscure journals. But basically, his work provided the genetic basis for Darwin's ideas about evolution, which have now been substantiated and are a part of how we do biology. He provided what's called the chromosome theory of inheritance. So he basically came up with this idea of chromosomes and genes that get passed on via chromosomes. And I know we talked about chromosomes with our unit on mitosis and meiosis. So let's get more into what Gregor Mendel did. He worked with pea plants, and one of the big experiments that he did was look at the flower colors of the pea plants. He noted that some of his pea plants had purple flowers and some of his pea plants had white flowers. And for a period of time, he isolated these plants based on their flower color so that he established in his garden, by blocking off certain areas, a purebred purple flowering pea plant population. There's some alliteration for you. He had one group of pea plants that only had purple flowers, and he had that repeatedly enough that he was certain that this is now a purebred line of purple flowering plants. And then he had another group of plants that produced white flowers only, and that was his purebred line of white flowering plants. So he first had to establish these purebred lineages in his garden. Then he did an experiment where he crossed a purple flowering plant, or actually hundreds of purple flowering plants, with white flowering plants. So a cross between purple flowering plant and a white flowering plant. And the result of that was that this next generation of plants, you could call these hybrids between the purple and the white flowering parents, this next generation was entirely purple. Okay. Interesting, huh? So what happened with that generation, right? Why is the white flower color not showing up in this generation of plants? So then Mendel did more experiments. He took the plants from this generation that's called the F1 generation. This is the first generation of offspring here. Plants within this generation, each of which had a white colored parent and a purple flowering parent, and crossed them with each other. So they bred together. And what did he get in the next generation? In that F2 generation, the second generation after the original, he had, and this is where he had almost 1,000 plants at this point that had been resulting from these experiments. And of those plants, 3/4 of the plants had purple flowers, and 1/4 of the plants had white flowers. So we've got a ratio of 3-to-1, or you could say 75% purple, 25% white. And just looking at that, I don't know about you, but I don't think that I would say automatically, oh, cool, I totally know how these traits are inherited now. But Mendel was a scholar that studied statistics. He studied math in addition to botany, biology, and he was able to apply the rules of probability and what he knew about statistics and come up with an explanation for how these colors resulted from these crosses. So this is what Mendel did. He said, okay, I know the results of my experiments, seeing the outcome in these traits, but what does this mean genetically? Mendel said, let's say that every individual has two versions of this flower color gene. Okay? And we're going to call these different versions of the gene alleles. And in this population, if flower color is a gene, one version of that flower color gene could be purple. And purple is going to have this big P here. And one version of the flower color gene is white, and white is going to be represented by the little p. And in those purebred lines, the purebred purple flowering plants had only purple alleles for this gene. So each of those plants had two big P alleles. And the purebred white flowers had only white alleles, so each of those had two little p alleles. Now, the reason these are big P and little p is because that big P, the purple allele, is dominant over the white, as you can see in this next generation, because if you cross a purebred purple plant with a purebred white plant, all of the offspring end up purple. So Mendel proposed that each individual has two copies of this gene, and their offspring get one of those copies. So a parent that has two big P's will give all of its offspring a big P. A parent with two little p's gives all of its offspring a little p, and in the offspring, they get one from each parent, which means in the second generation, all the individuals had one big P and one little p, and they were all purple. Remember that generation, everybody was purple. And he said that they have a big P from their purple parent and a little p from their white parent. The big P is dominant over the little p, so it gets expressed in this situation, and all the plants are purple. But when these plants reproduce, they are going to give half of their offspring a big P and half of their offspring a little p allele. And that is going to result in a more complex inheritance pattern. And here Mendel applied what we would call now a Punnett square. And he said you can put these alleles that are possible from one parent at the top of this square. So this parent could provide sperm, the male parent, it could be a big P allele, it could be a little p allele since they have both. And the female parent also could provide a big P allele or a little p allele. And you look at how these alleles can go together in various combinations, and that results in the potential for the offspring. So you can see here that 3/4 of these potential offspring are purple and 1/4 is white. Let's get into this a little bit more here. I'm going to define some terms and we're going to work through this together as well. This square is called a Punnett square. The genotype is the combination of alleles that an individual has, so it's the genetic makeup. The phenotype is the trait expressed. In this case, this is going to be purple or white. We can also have homozygous and heterozygous. Let's move into that. Okay. To repeat, a genotype is the genetic makeup, the combination of alleles. And recall that alleles are alternative or different versions of the same gene. In this example, the gene is flower color. The alleles are big P for purple and little p for white. The phenotype is the trait expressed, purple or white in this case. If an individual is homozygous, that means they have 2 copies of the same allele for a given gene. If they have 2 big P's, they are said to be homozygous dominant in this example. 2 little p's in this example, that is homozygous recessive. The recessive allele is the allele that is not expressed if there's a dominant allele present. And if they are heterozygous, hetero means different. So this means they have 2 different alleles for a given gene. And an example of a heterozygous genotype is big P, little p. And P's are not always used. We'll see different examples. Other letters are used for other traits. This is just for these flower color traits. And dominant refers to an allele that, if it's present, will be expressed in the phenotype. It will determine the trait. So let's go through this example of what Mendel applied together and say that in that generation, the first offspring generation, the F1, where all of the individuals were heterozygous. They were all big P, little p, and they all had purple flowers. When he bred those together, he could do a Punnett square and say, okay, one parent up here is heterozygous, so half of their offspring are going to get their big P allele, and half of their offspring are going to get a little p allele. So we're going to go down here to, see, I have to escape from this so I can put these in so you can kind of see me putting them in here. We're going to go into this Punnett square and put the big P allele from this parent one into these 2 squares on the left, and the little p allele from parent one into the square on the right. If you're following along at home, I hope you're drawing this Punnett square and you are working out how you would put these alleles in. So half of their offspring, it's like flipping a coin if the offspring is going to get that big P allele or the little p allele from their parent. And then from this other parent, parent 2 over here, they are making a contribution of 1 allele. So we'll take this big P here and put that in the top row. It's convention to put the dominant allele first if you have a dominant and a recessive. And then we're going to take this little p from parent 2, and half of its offspring get that. So we'll put that down here in the bottom row. And now we have a table that has the potential genotypes of the offspring. And let's just see if we can fill in those phenotypes. Well, maybe I'll start with the genotypes. I'll start with the offspring genotypes down here. These are the possibilities. So 1 out of 4 of these offspring are going to be homozygous dominant. 1 square out of 4. 2 out of 4, or you could reduce that to 1/2, are going to be heterozygous, big P, little p. And 1 out of 4 are homozygous recessive, little p, little p. I'm going to change this middle one to 1/2 so I can get a nice fraction there. And I've also reduced this ratio down so we could say that there's 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. That's the ratio of the expected genotypes of the offspring. And these are just expectations based on probability. So based on probability, if these parents had 4 offspring, these would be the predicted genotypes. But in reality, it's not determined what which allele the offspring are going to get. So basically this means that that offspring has a 25% chance of being homozygous dominant, a 50% chance of being heterozygous, and a 25% chance of being homozygous recessive. And then for the offspring phenotypes, I've colored in the background here to indicate that these 3 genotypes correspond with the color purple. That's the phenotype. So for the offspring phenotypes, we're going to have 3/4 purple and 1/4 white. That's what we would expect based on probabilities. And Mendel was able to look at his outcomes and deduce this type of relationship. And this is a Punnett square. We're going to do more Punnett squares in lab this week. So wrapping up a little bit about what Mendel proposed, some of this groundbreaking information here about heredity. Number 1, there are alternative forms of genes, and he called these alleles. These are called alleles. These alleles provide variation within individuals and within populations, and organisms inherit 1 allele from each parent. So for each gene, you get 1 allele from your mom, 1 allele from your dad. Any individual can be homozygous, having 2 copies of the same allele for that gene, or heterozygous, having different alleles for that gene. And dominant alleles determine the trait that is expressed. They determine the phenotype. If there's no dominant allele present, then a recessive allele can be expressed. Recessive alleles are only expressed in the absence of dominant alleles, as we saw with the white flower color, that little p. So recessive alleles are expressed in the absence of dominant. And I want to relate this back to what we learned when we did our lessons on meiosis. Recall that when gametes are formed, sperm and egg, there are pairs of homologous chromosomes that are separated. And these homologous chromosomes actually hold the same genes, and they hold one allele for that gene. So if you had a chromosome, let's say up here from mom, and that chromosome had a big P allele on it, and then a chromosome down here that you got from your dad, and it had a big P allele on it, then your genotype with respect to this gene is homozygous dominant, right? And you could have gotten a little a for a different gene, so this would be a different gene, different trait. Maybe you had a little a from your mom and a little a from your dad, so you are homozygous recessive for that one. And then maybe you're heterozygous for a different trait, a different gene. So these chromosomes have many genes on them, but those genes are going to be at a particular site on the chromosome. So for the homologous chromosomes, they're going to have the same genes, but they might not have the same version of those genes. They may not have the same allele. When this individual reproduces, half of its gametes are going to get this chromosome up here, and half of its gametes are going to get this chromosome down here. So it's going to pass on one allele for each gene to its offspring. And this is how we get this great diversity of offspring, because you have so many different combinations of these alleles that can result. Mendel applied a couple of laws to his observations here. One of these he called the law of segregation, and this simply states that these pairs of alleles separate to form gametes. Each sperm or egg that are produced only gets one allele from each gene from that parent. Okay? So that just means the parent is not passing on both alleles for a given gene, it's only passing on one. And that's really important because the offspring have to have two, but they get one from their mom and they get one from their dad. And he also applied this law of independent assortment, which we saw when we were studying meiosis, which states that pairs of alleles segregate independent of each other. And what this is really saying is that these chromosomes segregate independent of each other when they line up at the center of the cell for meiosis 1. When we talked about this law of independent assortment already, right? So when we have this line of homologous pairs of chromosomes, which one's going to be on which side, like the one from your mom or the one from your dad when they line up? It's random. It's like flipping a coin every time where those chromosomes are going to end up, which side. So you have a great diversity of gametes possible because of the law of independent assortment. And this also translates into how traits are passed on, because you have these pairs of alleles that also segregate independently during gamete formation. Mendel was able to get at this by breeding the pea plants again, and to elucidate whether traits segregate together or independent of each other, whether they are passed on as a group of traits or as each individual trait. He looked at the seeds of pea plants. And guess what? The seeds of pea plants are called peas. So he was looking at peas, and he was looking at their shape and their color. And he had variation in pea shape and pea color, and he was able to breed the plants enough to get purebred lines of round yellow peas in one lineage, so that the big R is for round and the big Y is for yellow. Those are the dominant traits for shape and color. And he also had another purebred line of wrinkled and green peas. So the wrinkled were the recessive, the lowercase r, and the green were recessive, and they have a lowercase y because yellow was the dominant. And when he did the experiments where he crossed those together and he got the next generation, which was all round and all yellow, he wanted to see then, if he did this cross with these heterozygous individuals, if he could get some peas that were round and green, and if he could get some peas that were wrinkled and yellow, or if these traits would always sort together and you couldn't get a wrinkled yellow pea. But what he found was a ratio that was predictable if those traits assorted independent of each other. So he got some round yellow and some round green and some wrinkled yellow and some wrinkled green. And that was his support for this idea that the two traits aren't going to segregate together, that for each individual trait, you can get whichever allele is passed on independent of others. Okay. So I'm going to move on into some pedigrees. And then we're going to come back a little bit more to some of these ways that traits could be inherited. But in our genetics lab, we are going to do a series of pedigrees. And a pedigree is a family history of a trait. We can use what we know from Mendel's experiments about how alleles get passed on to help deduce what alleles or what genotypes individuals in a family history have based on what we know about other individuals in their family. So let me just show you how to read a pedigree. In this pedigree, this is a family of individuals. Males are squares. Females are circles. A horizontal line connecting a male to a female indicates that they have mated. So this is a father and a mother pair. And this vertical line connects that pair down to their offspring. If a line goes up, across, and down, then it's connecting siblings. So we've got, in this case, dad, mom, can't get my cursor over mom, and then their daughter and their son. The pedigree is going to be for a given trait, and any individual in the pedigree that has that trait is going to be shaded or have a dark color filling in their circle or square. So in this pedigree, there's only one individual that has the trait, and it's this male right here. And the trait for this pedigree is deafness. It's the deaf trait. There are different causes of deafness, several different causes, but in this particular type of deafness, it is caused by a recessive allele being expressed. If an individual does not have this dominant allele, this dominant D, then they express the recessive little d genotype and have deafness. So again, down here, this individual who is deaf, who is a male, he's connected by a horizontal line to his partner, his mate, this female over here. What can we fill in based on what we know? Let's go back down to where we can fill things in here. So I know based on this, because I know right now that deafness is a recessive trait, I know that this individual, in order to express it, cannot have a dominant allele. I'm going to fill in his genotype as homozygous recessive. I also know that everybody else in this pedigree who has normal hearing must have at least one dominant allele for this trait. So I'm going to go in, I'm not going to fill out everybody in this pedigree, but these 5 individuals we'll look at. And I give them all a dominant allele, but I know they have to have another allele too. So can I fill in that next allele? Think about that. Mom and dad up here. Do mom and dad have big D, or big D, little d? Are they homozygous dominant or are they heterozygous? Giving you time to think about that. Noting that their son down here has 2 recessive alleles. He had to get one of these recessive alleles from his dad. So his dad has to have a recessive allele. We'll go ahead and fill in that little d. And his mom also has to have a recessive allele because he got a recessive allele from each parent. That is where his alleles came from. Right? So now down here we have daughter, and we know that she has a dominant allele because she is not expressing deafness. She has normal hearing. And we know that she has one big D. But what did she get for her other allele? Could it have been the recessive allele? Sure, if you do a Punnett square of mom and dad, it's going to turn out much like that Punnett square we did before, where you would expect 1/4 homozygous dominant, 1/2 heterozygous, 1/4 homozygous recessive. So she could be homozygous dominant or heterozygous, whoops. And I don't know, we don't know for sure which one she is. So we have to put a dash there for that second allele. And that dash just means we're not sure. We're not sure whether that second allele is a dominant big D or a recessive little d. We don't have enough information. If we had more of a pedigree here and she mated with an individual and then they had offspring, then we could maybe deduce more about her genotype. Likewise, over here with this female, we know that she has normal hearing. We know that her parents both had normal hearing. We know that she is mating with this individual who is deaf, but we don't know if that second allele for her is dominant or recessive. So we have to put a dash there. And this is something you're going to be working through in your lab on genetics as well. So let's see what this whole pedigree might look like. Here you see an extension of the pedigree, and we have more things filled in. We still are not able to know what the full genotype is for this daughter here, but if we extend this pedigree and we see the 7 kids from the individual that had deafness, that male, and his mate, then we see that some of their offspring have deafness, that they express that trait. So that makes us know that the mother had to be heterozygous because these offspring that are homozygous recessive, that have 2 little d's, they got one of those from their mom and one of those from their dad. So we try to fill in the information that we know, and if we don't know for sure, then we have to leave part of it blank, and I put a dash here. It's denoted as a question mark, but that just means we don't know what that second allele is for this individual. In a pedigree, you're going to see carriers, and carriers are individuals that have one copy of a recessive allele, but they do not express it. So you don't know that this individual, that this female, is a carrier of this deafness allele. And perhaps when she married or decided to have kids with this guy who is homozygous recessive and deaf, they didn't know if their offspring were going to have the risk of being deaf as well. They wouldn't know unless they had done some kind of pedigree in the past or other genetic analysis that revealed the genotype of this individual who is a carrier. So this is what a lot of genetic counseling is about, actually. For some diseases, they are so devastating that parents would want to go to great lengths to make sure that they do not have children with such a disease. Tay-Sachs disease that we talked about with respect to lysosomes is an example of that. So it's possible that 2 parents who don't have the disease, that are just carriers of the disease, could breed together and then produce offspring with a disease. And they might want to know in advance if they're both carriers so they can make decisions about reproduction based on that information. And that's where genetic counseling and testing can come into play. Okay, so to reiterate about a carrier is an individual who has one recessive allele. So they have it, they're carrying it, but it's not expressed. It doesn't affect their phenotype. In this case, with the deafness allele, the carrier would be the heterozygote. with normal hearing, but able to pass on that recessive allele to their offspring. Let's look at some common genetic, and these are strictly kind of a dominant recessive relationship, genetic disorders that we see in humans. And I'm going to start with some autosomal recessive. An autosomal refers to those chromosomes that are not the X chromosome or the Y chromosome, right? The non-sex chromosomes. So these chromosomes can hold, the autosomes hold lots of genes, and sometimes those genes can be associated with certain diseases. For example, cystic fibrosis. And these are all recessive, by the way. So the ones I'm listing here are recessive disorders. You have to have 2 recessive alleles to express them. Cystic fibrosis is the most common lethal genetic disease in the United States. It results in death in early childhood often. I mean, there are some individuals who can live into their 20s, but usually due to respiratory mucus buildup and infections, not a long life is able with this disease. It also mostly affects people with Caucasian ancestry, and we'll see how these recessive disorders do tend to be associated with certain areas of the world, right? With ancestry from certain geographic areas. And that's because these recessive alleles popped up in a population somewhere in a certain area. And if they were in that area for a long time, they just have not spread across all populations and aren't distributed evenly at this point. So you'll see them associated with certain biological heritage. So cystic fibrosis is most common in Caucasian populations that coming from parts of Europe. And then we have sickle cell anemia that is most common in people with more recent African ancestry. Sickle cell anemia is a disease that results in a change in the shape of the hemoglobin that results in the change in the shape of red blood cells. And these red blood cells then are not able to efficiently carry oxygen. So people with sickle cell anemia also have lots of health problems associated with that because they can't get oxygen to their cells efficiently. Tay-Sachs disease, we already mentioned. This is a disease that is most common in Ashkenazi Jewish populations. So people who have Ashkenazi Jewish ancestry, they will, in many cases, when getting married and deciding to have children, if they know that this Tay-Sachs allele is present in their family history, they might go and get a genetic test to see, okay, so if we have kids, do our kids have this possibility of getting this disease? And that's the disease, by the way, Tay-Sachs, that is due to an inability to break down fatty layers in the brain during development as a baby. So as an embryo and then as a baby after being born, usually resulting in death at a very early age. Tay-Sachs is a very devastating disease. We do have some autosomal dominant disorders. So these are the ones that are caused by an allele that if you have that allele, it's going to be expressed and you're going to get this. Ooh, let me get my pictures up here. Sorry. You're going to get that condition. By the way, this picture up here that's on the right, this is of a sickled red blood cell, typical of sickle cell anemia. And this is what a red blood cell should look like. It should be nice and open and able to carry a lot of oxygen. Well, I'm going to mention 2 conditions that are dominant. One of them is a type of dwarfism that is called achondroplasia. And if you're familiar with the show Little People, Big World, you might be familiar with this family. This is Amy, the mother, Matt, the father. Amy's form of dwarfism, achondroplasia, is a dominant autosomal form. So she had one copy of the dominant allele, and they had 4 children. You would expect about half of their children, on average, to get that dominant allele from their mother. But actually, what percentage of their children got that allele? It was 1/4, right? So again, these are just probabilities. Each time an allele gets passed on to a gamete, a sperm or egg, it's like flipping a coin. Which one's going to get passed on? The father, Matt, his form of dwarfism was recessive, or is recessive, and Amy was not a carrier of that form of dwarfism. So none of the offspring could get the form of dwarfism that their dad had. However, like I said, one of their sons did inherit their mother's dominant dwarfism allele. And another autosomal dominant, in this case disorder, is Huntington's disease. And Huntington's disease is a degenerative nervous system disorder. It usually shows up at middle age. So you wouldn't know that you have the Huntington's disease dominant allele until maybe 40s or 50s, when you start experiencing symptoms associated with degeneration of nervous tissue. Usually individuals who have Huntington's disease die about 10 to 20 years after the onset of symptoms, and they die from this disease typically. So if you have a parent who starts exhibiting symptoms of Huntington's disease in their 40s or 50s, you know that you, as an individual, have about a 50% chance of also having that allele that's going to cause that disease. And this can make you wonder, would you want to know? Would you want to get tested to see, do I have a dominant allele and I know is going to be expressed, and I know I'm going to get Huntington's disease, and that will likely be how I will die? That's a question of these genetic, knowing and understanding the genetics that we have. Do we want to know? In many cases, maybe yes, maybe no. I had a good friend whose father had Huntington's disease, and she and her sister made different decisions. One of them wanted to get tested for Huntington's disease, and the other did not. Unfortunately, the one who was tested did not have the lethal dominant allele. But again, people approach these things, and whether they want that information or not, differently. So I have a thought question for you, and I want you to think about this before our review session at the end of this week, in this case on Thursday. Dominant lethal alleles are not very common in populations. They're much less common than recessive lethal alleles, right? There are not very many deadly disorders that are caused by dominant alleles. Why? Why are recessive lethal alleles much more common? Look for that answer in our review. Okay, now we've been presenting everything as if it's very straight up dominant recessive, and that is not always the case. There are more complex inheritance patterns than those that are strictly dominant or strictly recessive expression patterns. For example, we can have incomplete dominance. And incomplete dominance means that the heterozygote is an intermediate. It's kind of like a blend between the phenotypes of the two homozygotes. An example with this carnation here would be if we have the red allele and the white allele. Then the red allele is a capital R, and the white allele is a lowercase r. The homozygous dominant carnations are bright red. The homozygous recessive are white, but the heterozygotes are pink in this case. So that is an incomplete dominance. The red allele is getting expressed, but it's not getting expressed to the same extent in the heterozygotes as it is in the homozygous dominant individuals. So this is really handy, because I can tell just by looking at these carnations based on this inheritance pattern what their genotypes are. I can tell the heterozygotes because of the incomplete dominance. With a codominance relationship, this means that both these alleles that are present are fully expressed in the gene, in the offspring, in the phenotype. So an example of this is our blood types. Let's see, a picture of different blood types. We can have type O blood, and with that O blood, it's these two I alleles, and the I alleles are recessive alleles. But A and B alleles in this blood type system are each dominant. And when you get one A allele and one B allele, they are going to be codominant, which means if you have an AB blood type, I'll wait for that little bar to disappear. Okay, the AB blood type at the bottom, that each of those are expressed. And that means this individual is going to make A carbohydrates and B carbohydrates, which is what these blood types are about. They're about carbohydrate production in the blood. There's an example from the genetics lab about codominance, and the example in the genetics lab is a tortoiseshell cat or a calico cat, an expression of black and orange colors in those cats. They're codominant. They both get expressed. Then we have a condition called pleiotropy. And with pleiotropy, one gene affects many traits. And an example of that would be a gene that's involved in turning something on or off early on in development. We did talk about a gene last week that was, or in our last lectures, that was this gene that's on the Y chromosome that says be male, right? That changes the course of embryonic development. The default development is female. But if that gene is expressed, then testosterone gets cranked up, and that causes the development of testes, and the embryo then develops as a male. So if a gene gets turned on in early development, it can affect a suite of traits, right? Think about all of these traits that are involved in being male. Those could be expressed by that single gene. Sickle cell anemia is another example of this. We have the hemoglobin gene that determines the shape of the hemoglobin. That affects the shape of the red blood cell. And sickle cell anemia, because it's involved in carrying oxygen, can have an effect on a suite of other traits. Like it can lead to brain damage because not enough oxygen is getting to the brain. It can lead to kidney failure, heart failure, and other health problems. So that's this one gene that's affecting many traits because of its mechanism of action. Polygenic inheritance. In this case, poly means many, poly, and genic genes. This is when many different genes influence one single trait. And examples of polygenic inheritance in humans are our skin color and our height. And I want you to think about human skin color and human height. These are not discrete traits. There are some people who might say, oh, I'm tall. I'm definitely tall. Or I'm short. I'm definitely short. But a lot of people are going to say, well, I'm in between. Or, you could say my skin is dark, or my skin is light, or my skin is very, very dark, or my skin is very, very light, or my skin is kind of a medium shade. It's not very dark, not very light. There's a wide range. So we can't say that you are this or not this, right? You are part of a wide range, ranging from one extreme to another extreme and everything in between. And this is an example of how skin color could be inherited in humans. And this is not actually how it is done, but there are multiple genes involved. So if you could just say, well, let's say that an individual at one extreme has these multiple genes, there are only 3 genes here, and has the very light alleles for each of those genes. And another individual has the very dark alleles for each of these genes, and they breed together, and they have offspring who have an intermediate skin color. Right? And then if these offspring breed, look at all these combinations. just from 3 genes involved. Now, granted, these are 3 genes, and this is an intermediate inheritance, incomplete dominant situation. But you see here this wide range of possible skin tones that resembles the wide range of skin tones in human populations. So you've got this variance. And height is another example, right? You've got very, very short people, very, very tall people, and then a wide range of heights in between. So that's an indicator, that wide range of traits and kind of continuous traits from one extreme to another extreme with a lot of individuals in between. That's an indicator that that trait is influenced by multiple genes working together to determine the phenotype, polygenic inheritance. And finally, the environment affects traits. And I put here, what is the environment? Well, the environment is everything around you. It is who you interact with. It is your other genes and what's being expressed and what kind of foods you can eat and so forth. It's not just the temperature or whether it's raining or not, but it's anything that you're experiencing in your world. That is your environment. And we have this idea of nature versus nurture, right? So nature, we say, is our genes. This is what we have, that we are born with, that we can't change. It's just part of who we are. And we're going to express those genes and make certain proteins. But nurture is our environment that begins even in the womb as we're developing as embryos. And that environment continues throughout our lifetime, that we're constantly being influenced by what's going on around us. And it's a combination of our genes and our environment that ultimately determines our traits. And this is called the gene by environment interaction. And what this means is our genes affect our traits, and our environment affects our traits. And also, our genes and our environments interact to affect our traits or our phenotypes. For example, your genes are going to affect your environment to some extent, because your genes might cause you to or influence the way that you react in a certain situation, right? Maybe your flight or fight response. And that fight or flight response is going to potentially affect the way that other people react to you. So that's going to give a feedback from your environment, because other people are your environment. Your environment can also determine to some extent which genes get turned on and which genes get turned off. So you can have substances that are coming into your body. Maybe they're based on what kinds of foods you're eating, how much energy you're getting, whether you are malnourished or having too many calories coming in. Those can trigger certain tags being put on your DNA, and it can actually influence which genes get expressed at certain times. So these two interact with each other. They don't just work independently. I want to show you some examples of how the genes in the environment might be both influencing a trait. So we have this individual who has clearly been out in the sun for a while, and we see the tan line here. So this individual has a baseline skin color that is largely determined by genes. But having been out in the sun and exposed to that light, that has influenced the proteins and changed the proteins to some extent in the skin, at least temporarily. And this individual now has what you could consider a tan or a burn, and that's the environment affecting the skin color in this area. So you can kind of see nature, the baseline skin tone, skin color, plus the effects of the environment of being out in the sun. With respect to height, I look at this graph here and I see that the height in inches of Major League Baseball players has been determined over time from the 1800s, the late 1800s, up into the early 2000s. And it's also charted with the average height of in the United States, and I assume this is of males of a certain age that would be roughly the age of Major League Baseball players. So you can see that you have your height of hitters, you have your pitching height, and those have steadily increased over time. And also, using as a control just the US population, the green line, that our heights as a population have increased over this time too, maybe not as dramatically as that of Major League Baseball players. So is this genetics? Is this like selection for genes that are making people taller and taller and taller? Or could we invoke some kind of environmental explanation for this? Let you think about that for a minute. And I think because you see this increase across the board, and think about what's been going on in the United States since the 1870s. We have access to more nutritious foods, perhaps, to a more balanced diet, to more fresh fruits and vegetables throughout different times of the year. And it's likely that nutrition has played a big role in these height increases as an average. So we would look for these environmental explanations as well as genetic explanations. Now, our population is also just a heck of a lot bigger now than it was in the 1800s. So we have more individuals, which means more variation, which means more height extremes. And so maybe you just have more tall players to choose from, and tall players might have an advantage on the playing field. So you'd look for these different explanations for the traits that are being expressed. I'm going to end this lecture now, and I'm going to leave a discussion of sex-linked traits for a future lecture. So look for that one, but this is the bulk of our lecture on genetics and heredity. Thanks much.